Hello, good morning, everyone. I'm going to go ahead and get started and do the test review. For you, there has been a little bit of problem with accessing the file over the weekend, but I sent out an email to everybody and redirecting you to the review sheet. Just in case, I'm going to put it in the chat box and you can click on it if you haven't already downloaded a copy. If you have any questions, just unmute yourself and ask away or type it in the chat box. But here we go. So first section is the microscopy lab, and this is the first question. Describe the relationship between the object and the image. Draw the image of HEP and viewed under the microscope. So the relationship between the object, which is what you place on the slide, and what you're looking at, the proper way of describing it would be inverted. Okay, inverted means rotated 180 degrees. Draw the image of H, E, and P. If you are stuck on the test and you're not sure how to draw it, the best way is just draw it normally, and then you physically rotate your paper, okay? So you rotate your paper and then you rotate your paper again, and then that's what it's going to look like. All right. So similarly, for the E, it would be like this. And for the P, it would be like that. Okay. So those are the image that you will get when you rotate H, E, and P. All right, number two, explain how total magnification is calculated. Okay, so total magnification is going to be the ocular lens magnification, which is by default 10 on most microscope, multiplied by the objective lens. Objective lens are the lens that you can rotate at the bottom. If you remember the microscopy lab, there are 40, there are 4, 10, and 40, right? So 4, 10, and 40. So you just multiply both of them, and you're going to get your total magnification. All right? So that's pretty straightforward. Okay, the numberings are all wrong when I copy it over, but it doesn't really matter, I guess. I'll try to renumber them. Okay, a ruler is viewed under the microscope. Find the diameter of each field. The key thing to remember here is the spacing between two lines. And the spacing between two lines of a ruler is 1 millimeter. So you just have to count across the middle of the field. Okay, don't count here. All right, we're looking at the diameter. The diameter of a circle is going to be right across the middle. So here you can see 1, 2, 3, 4 and a bit, 4.5, we can say. So that's 4,500 micrometer. Okay, yeah, I mean you can do 4.5 millimeter first, and then times 1,000 to convert it into micrometer. So same thing over here. 1, 2, 3, 4, 5. I can see the sixth line here, right? So 5 spaces, which is 5 millimeter times 1,000. That would give you 5,000 micrometer. And finally, over here, we have 1 space, 2 space, and a bit. And I mean, this here, I don't really see the next line, but judging from the distance here, I would say this is about two and a bit, 2.2 millimeter maybe, or 2200 micrometer. You're up with 5.0 millimeter here. Okay, that's how you figure out the diameter of the field of view, DFOV. Okay, next question, number 4. The DFOV at 40 times magnification is 2500 micrometer. What is the DFOV? Sorry, what is the DFOV at 400 times magnification? So we just use the formula DFOV of number 1 times magnification number 1 equals to DFOV number 2 times magnification number 2. So the first DFOV is 2500 micrometer. Please bring a calculator for the final test. Magnification is 40 times, okay, which equals to the new DFOV multiplied by the new magnification. Okay, so to solve for this, it will be DFOV2 equals to 2500 times 40 divided by 400. So plug that into the calculator if you need to. But you can reduce, and this is 10. And then in the end, you're going to get 250 micrometer. Okay. Yes, this is being recorded and it will be posted at the usual place, the archive, right? After class. I'll post the filled in notes as well, if you don't want to watch the whole thing again. All right, that's that. That is number 4. Number 5. The DFOV is 500 micrometer. So it's already given to you. From one end to the other end, it is 500 micrometer. And we want to find the length of the cell. So that's one cell, and roughly you can see you can fit another cell here, right? So that means the length is going to be 500 micrometer divided by 2, because you can fit 2 cells. If you can fit 2.5, then you divide by 2.5. If you can fit 4 cells, then you divide by 4, right? And that's going to be 250 micrometer. That's all you have to know for the microscope stuff. And all the microscope questions are going to be in the multiple choice. All right. Any questions? Great. Do we need a ruler for the test? No. Well, no, I don't think so. No. Yeah. All right, next part. What is the correct model for DNA replication? Right, so there are 3 proposed model, right? The correct model is semi-conservative. Semi-conservative. Okay. Which means if you have this as your original DNA, afterwards you will have half a strand, or one half of the DNA, it's going to be the original material, and then you're going to have the other half consists of new material. All right. So this is semi-conservative. You do not need to know the experiments for figuring this out. Okay, you just have to know the correct model and what it means. Okay. Oops. Name 3 differences in DNA replication between prokaryotes and eukaryotes. So everything we learn in class with the, ligase, helicase, DNA polymerase 3, and all that stuff, those were done in prokaryotes, right? Those were done in E. coli, for example, and we are eukaryotes. So there are some key differences between what's happening in the bacteria compared to what's happening in us. And these 3 things are written in the PowerPoint slides. Specifically, there are multiple origin of replication in eukaryotes. Okay, which makes sense because eukaryotes have a lot more DNA to copy, so you need to open up multiple bubbles to speed things up. A second difference is that the same enzyme has multiple versions in eukaryotes. Okay, so in prokaryotes, there are DNA polymerase 1 and 3, but then for eukaryotes, there are 5 different types of DNA polymerase. Right, so they do similar things, but there are just like multiple versions of it. And the last difference is that because eukaryotic DNAs are linear as opposed to circular, some DNA, well, we'll be more specific, DNA located at the ends of chromosome are lost after each replication. Okay? We'll say some. Okay? Some DNA located at the ends of the chromosomes are lost after each replication. And that's because of the way DNA polymerase 3 works. It can only go in one direction. So if you remember, let's say this is 5 prime to 3 prime. And of course, on the other strand, it would be the other way around. So you're gonna need to have like a primer to start building your new DNA strand. Right? So these are the primers, and then you're gonna build a new strand, build the new strand. As you approach the primer, you'll replace it with actual DNA. But the problem is this portion here, that is not replaced. Okay. Because there are no DNA polymerase coming from above, from before. This is the end of the line, right? And as a result, you lose a bit of DNA every time you copy. Same thing is true for the bottom strand. Okay. Same thing is true. You're going to be able to copy in this direction. And then here is not replaced with DNA, and it's going to be lost. The primer is RNA, right? As you might remember. Next, draw a series of pictures to show the process of mitosis. I'm not going to ask you to draw on the test, okay? But I am going to show you pictures of mitosis, and you will have to know which one is which. Right? So make sure you can identify the pictures So for example, this is prophase. Okay, prophase, the chromosomes are like, sorry, I drew it wrong. The chromosomes are already doubled and paired up, and then you would have your spindle fibers coming out. After prophase, it is metaphase. And for metaphase, the chromosomes are lined up in the middle. After metaphase is anaphase. And here are the centrioles again. And in anaphase, the sister chromatids are separated and they're being pulled to opposite end of the cell. And finally, we have telophase. Which is where you will separate the two cells and you're going to start to form your nucleus again. Okay, and then you're going to cut here, separate the cytoplasm, and the term for that is cytokinesis. Okay, division of cytoplasm. Again, you don't have to draw this on the test, but you need to recognize what they look like. I'm going to show you pictures, and you have to tell me the right order. Okay, moving on. Homologous chromosomes. These are basically the paternal and maternal chromosome pair. They are same length, same size, and contain genetic info contain same type of genetic information. Okay, so chromosome 1 and chromosome 1, they are both homologous to each other. 4 is homologous to 4. 6 is not homologous to 8, right? They have to be the same number, basically. Alleles, you should know what an allele is by now. They are different version of a gene, big B, small b, right? Those are two different alleles. Of course, some alleles are dominant over the others. And then the one that is being covered up, we call it recessive. Some alleles have incomplete dominant effects over each other. So in the end, you might get like a blend of each allele, right? Running out of space here. Let me just press enter a couple more times. Autosomal chromosomes. Autosomal chromosomes are in humans. In humans, they are chromosome 1 to 22. Okay, excluding sex chromosome. All right, so they carry genes regarding everything about you except for the sex. Finally, sister chromatids. Sister chromatids are identical copies of DNA, of chromosomes, you can say, held together at the centromere. So let's say this is chromosome 9, and then you have DNA replication. Then afterwards, you're gonna have 2 exact copies. Okay, it's gonna be stuck together at the centromere, and each copy would be a sister chromatid. Okay, I'm gonna pause for a moment and let you catch up and ask questions if you have any. Okay, moving on. Explain the significance of telomeres and telomerase. How does this relate to aging, related to cancer, and related to Dolly? All right, so let's talk about telomeres and telomerase first. So here is a chromosome. Chromosome. And located at the end of the chromosomes are non-coding sequences of DNA. So that's a telomere. Right? Telomeres are non-coding repeats of TTA-GGG. Right? The purpose is basically to protect genes near the ends of chromosomes. Right. Earlier we talked about how every time you copy a little bit of a DNA, you copy your DNA, you lose a little bit of telomeres, right? So you're gonna chop, 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 chop like that, and eventually it's gonna be too short, right? Uh, and you start eating into the genes, right? So we don't want to eat into the genes. That's why the telomeres are there, right? It protects them. So what about telomerase? Okay, telomerase. It is an enzyme expressed in germ cells, which helps, which allows telomeres to re-lengthen. Okay. So that's necessary because when you make the sperm and egg, you want the telomeres to be reset before you pass it to the next generation. So the next generation will have brand new telomere length. Otherwise, if it keeps on getting shorter and shorter, shorter throughout your lifetime, and you pass on short ones, eventually it will be too short to to propagate, to pass on. So how does this relate with aging? Okay, so cells with critically short telomeres cannot divide. Okay, they will fail G1 checkpoint. Okay. The cell becomes senescent. Right? So if your body has um, a pool of senescent cells, more senescent cells, it contributes to the effect. It contributes to overall aging. Number two, how does this relate to cancer? Telomerase are usually overly expressed in cancer cells, granting them limitless replicative potential. It means they can keep on dividing and dividing and dividing and not stop, which is how a tumor forms, right? And number three, how does this relate to Dolly the clone sheep? Dolly was cloned from the nucleus of an adult cell with shortened telomeres because the adult sheep has been having cell division throughout its lifetime, right? So with shortened telomeres. This means Dolly was born with shortened telomeres. So genetically, it was born as an adult sheep. Okay? So that's why it died earlier than like a regular sheep would, right? Has a shorter life expectancy because even though like it was born today, but on a cellular level, looking at its DNA, it's as if it is an adult sheep already. Are we together, guys? Any questions? All right. Stay three purposes of mitosis. And mitosis is to for growth and development. Replace lost or damaged cells. And for some organism, asexual reproduction. Okay. And what is BRCA1? This is as you might remember, a tumor suppressor gene. That slows down the cell cycle. Okay. It is involved in repairing DNA damage. Okay, that's pretty much it. Okay, so another thing to remember is people who inherited one mutated copy of BRCA1 gene is at a significantly higher risk of developing breast cancer. Okay, that's that. Please pay attention to the exclusion list. These are things you do not have to study for. Okay, I told you this in class already, but every question that we are doing right now, like all these questions, every single one of them is going to show up one form or another on the test. Okay, this is all you need to study for this review package. Okay, all right, here we go. Next lesson, lesson 7. A DNA segment has 300 nucleotides. Give me a second here. How do I make this go away? Oh, there we go. A, how many how many amino acids does this DNA segment encode? What assumption did you make? Okay, so we know 3 nucleotides codes for 1 amino acid. So if you have 300 nucleotides, that's 300 divided by 3, which is 100 amino acid. Okay, but we made an assumption. Okay? We are assuming the DNA segment does not contain introns. Okay, remember, introns are non-coding sequence. That will be removed. Okay, so I guess what you can say is at most, if the DNA segment is 300 nucleotides, at most, you will have 100 amino acid, assuming it does not have any intron in it. If it has introns, then it's going to be shorter than 100 amino acid. Okay? Next, the 24th 24th nucleotide is deleted. How many amino acids will be unaffected by the deletion? Okay, so I mean, I'm trying to think of a way to explain this without drawing it out. 1, 2, 3, dot, 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 dot. 22, 23, 24, dot, 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 dot. So the 24th here, this is deleted. This is deleted. How many amino acid will be unaffected? Well, let's see. The first three here, that's going to be amino acid number 1, and then 4, 5, 6 is going to be amino acid number 2, right? And then so on and so forth. Right? So let's see, up to the 21st nucleotide, there will be 7 amino acid, right? 7 amino acids. And then 22, 23, 24 nucleotides would have been the 8th amino acid. But number 24 is deleted. So number 8 is going to be wrong as well, right? This is supposed to be the 8th nucleotide, remember. But if you change the last letter, if you delete it, then you're gonna have a frame shift mutation, and this is gonna be wrong. Everything else after that is gonna be wrong. So 24 nucleotides, sorry, 7 amino acids are going to be unaffected. Okay. It means number 1 to 21 are okay. So 21 divided by 3 is 7. That's basically how it is. How about this one? A nucleotide is inserted after the 50th nucleotide. Okay, so 1, 2, 3, dot, 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 48, 49, 50, and then you insert an extra one. Right? So how many will be unaffected? Can anybody give me a number? Okay, so this is the closest number that's divisible by 3. So from here, if you do 48 divided by 3, that's going to be 16 amino acids. Okay, and then this was supposed to be the next triplet, but then you insert something which makes it wrong. Right, so the correct answer is 16 amino acid. Okay, that's all. Moving on. Given the coding strand, here, write the RNA sequence. You should be pretty familiar with this by now. If it's the coding strand, all you have to do is copy it, but change the T to U. There you go. Translate the mRNA sequence, which means you are going to need to have your codon chart. I need to bring my up. Just give me a second. Of course, it's going to be given to you on the test. So right here, So AUG is MET. You should know that. That's a start codon. GAC, GA, and then look for C right here. That's ASP. CAA, CAA is GLN. UAG is stop. That's all. Okay, so create a nonsense mutation by base substitution. Okay, let me just take the original sequence here. I know this is CAA, so if I change it to TAA, then that's going to be a nonsense because TAA will become UAA, which is a stop codon. Okay, so base substitution means you only change one nucleotide. All right, let's take a look at insertion. How do we do an insertion to create a nonsense? Well, I can put it right here. If I put a T here, then it will be AUG and then UGA. And that would be a stop codon. Okay, so you can create a nonsense mutation which causes the protein production to stop prematurely, either by base substitution or by insertion. Actually, you can do it with deletion as well, although that's a little bit harder. Okay. Next one here. Oh boy, what am I doing? It's okay. The numberings are wrong, but that's okay. I'm not gonna fix it. Create a neutral mutation by base substitution. So pick any. This is GAC. GAC, it's right here, which codes for ASP. But GAU also codes for ASP. So that means I can change this last one to a T, and it would be neutral. Okay, so both GAC and GAU code for ASP. No change in the amino acid. That's a neutral mutation. Okay? You cannot have neutral mutation with insertion or deletion because you create frame shift, right? But with base substitution, it's okay. All right, let's take a look at this next one. Hemoglobin is an oxygen-carrying protein found in red blood cells. Its quaternary structure contains two alpha chains and two beta chains. Thalassemia is an inherited blood disorder that causes your body to have less hemoglobin than normal. In some forms of thalassemia, the beta chains are completely missing in some hemoglobins. What kind of mutation is the likely cause of this? Explain. So if it's completely missing, that's like the key thing here, right? If it's completely missing, then it's probably going to be a nonsense mutation. Why? Because a nonsense mutation causes the protein to be shortened, okay, truncated. Truncated. Okay. If the nonsense mutation can't spell this morning. Nonsense mutation. If the nonsense mutation happens early in the gene, it's possible that the entire protein is gone. which is what happens in the beta chain here. It happens really early, and you end up not making the protein, and then your hemoglobin is incomplete. That's it. Okay, explain the likely outcome of missense, nonsense, and frame shift mutation. All right, so uh, the severity of the mutation, like how bad is it, depends on the location of the mutation. Okay, so I'm just going to give you a couple of examples and you can think about the rest. E.g., a nonsense mutation near the end of a gene is not as damaging as one near the beginning or in the middle of a gene. If the nonsense is happening at the end, then you lose a few amino acids, and that might be okay. I'm not saying it's gonna have like no effects, but it's certainly not as bad as happening at the beginning, which you will end up losing the whole protein, or happening in the middle, which you lose half the protein. Okay, so happening at the end, it's not too bad. Okay, another example. I'm just gonna switch color here. A missense mutation happening in the active site of an enzyme is more damaging than one that happens elsewhere in the enzyme. Because active site is where you're going to fit the reactants. And if you change the amino acids in the active site, you might end up changing the shape of the active site, and it will cause the enzyme to not work, right? I'll give you a few other examples here. Missense, if it's like a hydrophobic amino acid to another hydrophobic amino acid, okay, then maybe it's okay. Maybe it's okay. But if it's a hydrophobic amino acid mutated to a hydrophilic amino acid, right, probably not okay. And frame shift is generally quite bad. Okay, it doesn't really matter where it happens. It's like it's bad. Right? If it happens at the beginning, very bad because the whole chain is gonna be wrong. If it happens in the middle, then again, quite bad. Half of it is wrong. But even if it happens at the end, it's still quite bad because look, you're gonna have like normal amino acids, and let's say you're supposed to stop here, right? But if you have a frame shift here because of insertion or deletion, then you will miss the original stop and you end up adding a bunch of extra things. And who knows how long will this thing be? Okay. So it's bad. Frame shift, beginning, middle, end. You don't want frame shift mutation. Okay, so just take into consideration these things when you are reviewing, okay? All right, moving on. Use a diagram to illustrate the following terms. Okay, let's move quickly. We still have a lot of things to do. If you have a gene, okay, this here uh, is the promoter, which is the beginning of a gene. This is terminator, which is end of a gene. And then within the gene, you're gonna have two kind of stuff, two kind of sequences, okay? All right, so you will have parts that will be expressed. These are the exons. They are coding. And in between the exons are going to be the non-coding. Non-coding. And those are the introns. Okay, which you will have to remove after transcription, right? Okay. So you're gonna go ahead and do transcription. And let's say you've removed everything already, so you only have the green stuff. To increase stability, you're gonna have to put a G cap here on the 5' end, and then on the 3' end, you're gonna put a bunch of A's. Okay, the poly A tail. Right? So that's where all the terms fit. All right, explain the relationship between codons, anticodons, and tRNA. This is 5' and 3', and this would be the messenger RNA. And on it, you would have a codon, UUU, for example, codon. So then there is going to be a tRNA molecule, which is responsible for bringing the correct amino acid to the ribosome. Right here is the ribosome. So how do we ensure the right tRNA is docking into the ribosome? Well, at the bottom of the tRNA, there would be a triplet of anticodon. The anticodon and the codon must be complementary. If they're not complementary, then it's not going to fit. So the amino acid that it's carrying, well, you have to look up the codon. Never look up the anticodon, okay? So you look up the codon. UUU, if you look it up, it's PHE. So that's the relationship between codon, anticodon, tRNA, and amino acid. Okay. Any questions? Okay. I'm gonna have to skip the break and keep going. All right. We still have how many? 3 more lessons to go? Yeah. 8, 9, 10. All right, heredity. Explain the significance of crossing over. How, why, where, and when does it happen? So crossing over is basically when the maternal chromosome pairs up with the homologous paternal chromosome. Okay, so that's the crossing over right here. Crossing over. Okay, how? Maternal and paternal homologous chromosome form tetrads. Form a tetrad. That's how. Right? This whole thing is a tetrad. Why? Exchange of genetic materials to increase genetic variation in gametes. Where? Happens in the gonads during gamete formation. When? Prophase 1 of meiosis. So remember afterwards, right? The red's gonna have a bit of blue and the blue is gonna have a bit of red, right? That's basically what. Okay, next. What is the purpose of meiosis? Where does it happen? What is the result? So meiosis is the process of gamete production. Okay. It generates four daughters, four haploi, four genetically different haploi cells. Okay, and I'm gonna put a note here. Four sperms in males. For female is one egg plus three polar bodies. Okay. Only the egg is a gamete. Right? These are not reproductive cells. Where does it happen? Testicles in man and ovaries in female. Next, what is non-disjunction? Non-disjunction is basically failure to separate chromosomes. During when? During anaphase 1 or 2. And what's going to be happening at the end? The result is gametes. Ooh, containing extra, so n plus 1, or missing n minus 1 chromosomes. All right, make sure you practice the monohybrid, dihybrid, all these questions, okay? Just we do the ones from the study guides, do the ones we have in the lecture, then you should be okay. It's not going to be more difficult than the ones we've done in class. All right. Obviously, go through the quiz number 4 as well, especially if you didn't do well, watch the review video for lecture for quiz number 4 and make sure you know how to do those problems. Okay? Next, how many unique gametes could be formed, could be produced through independent assortment by an individual with genotype da 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 da. Okay, so big A, small a, there are 2 choices. Big B, small b, 2 choices. Big C, 1 choice. 1 choice for small d, small d. If they're the same uppercase or lowercase, then it's only 1 choice, right? Big E, small e, 2 choices, 2 choices, and you multiply all of them together. Okay, so 16 in this case. 16 combinations. A cat with long tail breeds with a cat with no tail. All kittens or the babies have short tails. What inheritance pattern best explain this? So like if you have long and none, and then you get something in the middle, this would be incomplete dominance. Okay. Where the heterozygous phenotype is an intermediate blend, if you would, of the homozygous phenotype. So it's not quite like big L, big L. It's not quite like small l, small l. Is somewhere in between. Somewhere between long and none would be short. Okay. Brown eyes is dominant over blue eyes. Is it possible for 2 people with brown eyes to have a blue eye child? What do you guys think? Anybody? Been talking for like an hour and a bit. Anybody want to take over? Two people with brown eyes. Can they have a child with blue eyes? That's right, Angel. If both are big B, small b. Yes. If both parents are homozygous. Right, so like big B, small b, cross big B, small b. Okay, you guys got to be careful when you do the Punnett square, okay? On quiz 4, a lot of you got this right, but when you put it in the Punnett square, like you made a mistake. You put big B instead, and then everything is wrong. Okay? Please make sure you put the correct letters in. Okay, so in this case, there is actually a 25% chance for blue eyes. Now, I will put a star here. The opposite is not true. Okay? Blue eye parents, if both are blue eyes, is going to be 100% blue eyes, okay? So for recessive traits, it's not possible to have a child with something different, but with dominant traits, it's possible for the kid to be different if the parents are homozygous, for example. Okay, next one is a bit of a brain teaser. Janet has normal vision. So what does that mean? Normal vision means X something, right? Like we don't know what the other X is. Her husband has normal vision. Husband is guaranteed to be XY like that. Is it possible for them to have two sons where one has normal vision while the other is colorblind? Explain. Okay. We don't have a lot of time, so I'm just going to tell you yes, it is possible if Janet is a carrier. If she's XXC, right, then it's possible. Look, XXC for Janet, and for the husband is XY. So we are focusing on the sons. Right? You can see here for the son. This one is going to have normal vision. This one is going to be colorblind. Okay. Never mind the daughter because it's not asking about the daughter, right? So this is normal. This is colorblind. Okay. Is it possible for them to have two daughters where one has normal vision while the other is colorblind? Well, let's take a look. Okay, let's see if it's possible. So over here, uh, it's XX for one and then XXC for the other. And you can see they are both going to be normal vision. So no. Okay. I mean, we don't even have to try the other possibility, right? The other possibility is if Jenny is like XX, right? If she's XX, then of course nobody's going to be colorblind, right? So the other option is she's a carrier. But even if she's a carrier, then you can see everybody, all the daughters, going to have normal vision. Okay. We're not going to try XCXC because she has normal vision. It's impossible for her to be XCXC. Okay. In fact, if you take this a step further, a husband, a male, a father, we'll say a father, okay? A father with normal vision will never have a daughter that has color blindness, regardless of who the mother is. Okay? Because if the father has normal vision, the normal X chromosome will cover up any colorblind X chromosome. So it's not possible to have a colorblind daughter. Okay. Next, consider two traits, flower color, and number of petals. For the parent, they are pure. Okay, so please remember, I've told you this many times in class and we practice doing this. If the parents are pure, if the P generation is pure, then the genotype, the phenotype, the appearance of F1 will tell you which one is dominant. So here, orange flower and 6 petals. Okay. So that means orange, which I'll use R and not O. Orange is dominant over pink. Here, orange is dominant over pink. And 6 petals is dominant over 4 petals. You cross two F1 individuals. If there are 2,000 offspring, approximately how many will have orange flower and 6 petals? So orange flower is a dominant trait, and then 6 petals is also dominant. So if we're looking for dominant, I mean, you can use the table to help you, but really dominant is 9 over 16, right? And we're going to times 2,000 this time because there are 2,000 offspring. Plug this into the calculator, you will get 1125. Now, if you get something like this on the test, you must explain. Okay, if you just write all this stuff, I'm not going to give you full marks. You have to explain to me how did you know orange is dominant over pink? How did you know 6 petals is dominant over 4 petals? How do you know to use 9 over 16 and not like 3 over 16 or 1 over 16? Okay? You have to explain your work. Otherwise, not going to get full marks. Okay, moving on. This one. Consider the cross A. Okay, I'm not going to read it out. Okay, you can see it here. What is the probability of producing genotype of this? Now, it would be crazy to list all the possible gametes because look, this is 2 times 2 times 2, which is going to give you 8 gametes. And this is 1 times 2 times 1, which is going to give you, I guess not too bad. It's going to give you 2 gametes, okay? But still, it's a lot of work, okay? Like, what if this is, 8 choices and 8 choices? Your table will be like 64, right? So that's not the proper way of doing something like this, okay? Anytime you have more than 2 traits, you should not be doing a Punnett square, okay? It's just like too much work. So let's see. We want to have big A, big A. So what we're going to do is we're just going to look at the A here. We're just going to look at the A. So this is big A, small a, and then big A, big A. So big A, small a, and big A, big A. We're just going to do the A's alone. So fill in the table like you normally would. And based on this, what's the probability of big A, big A? Well, 2 out of 4, right? So that's 1 over 2. Next, we want a big B, small b. So we're just going to be doing the b's alone. A big B, small b. Here, I highlight them for you as well. We're looking for big B, small b. So that is also 50%. Okay, big B, small b, 1 over 2. And then lastly, we'll do big C, small c. So we'll just do the c alone. So that's simple, okay? Rather than doing a huge table. And we're looking for big C, small c. So that is also 1 over 2. So what's the probability? Okay, so I have 3 probabilities here. 1 over 2, 1 over 2, 1 over 2. Remember I told you in class, if you're in doubt and you're not sure whether to add or multiply, multiply is your best bet. Okay, and that's how you do it. Okay, to find the combined probability, you multiply the individual probability. So that's going to be 1 over 8. Okay? Please know this. So you basically do it 3 separate times, find the individual probabilities, and then multiply them together. Okay? Next. Hemophilia is an X-linked recessive bleeding disorder. Use the following information to create a pedigree. Okay, so let's see. Alice has hemophilia and she marries Terry. So there is going to be Terry and this is going to be Alice. Mary. So you're going to connect them like that. So she has hemophilia, which means I need to shade her in. And that means she is XHXH. C for colorblind, H for hemophilia, right? Alice and Terry have 2 daughters and a son. 2 daughters and a son. Okay. One of the daughters married a man. Okay, so I'll say it's this one. One of the daughters married a man, and they have a hemophiliac son and a hemophiliac daughter. Right? Hemophiliac daughter and hemophiliac son. That means this is a XHXH, and this is XH and then Y. What is the probability that their next son, right, their next son has hemophilia? Okay, so let's see what we can say about this. If the daughter is XHXH, that means one of the XH must have came from the mother, and then the other came from the dad, which means the dad must also have hemophilia. Okay. Now over here, Terry does not have hemophilia. He's normal, right? So he's X capital Y, which means this daughter must be a carrier. Okay. So we have enough to answer the question, but let's take a look at the other kids. This one also must be a carrier because the father will always pass on a normal X, the mother will always pass on the affected X. And then for the son here, son is gonna get the Y from dad. It's gonna get the XH from Alice because that's the only one she can pass on. So this one is also shaded. So here, what is the probability that their next son has hemophilia? So we're gonna do the Punnett square. Okay, we're only looking at the sons. 50%. If they have a son. Okay. Any question about the pedigree here? Really? I guess that's good. Okay, moving on. Answer each of the following questions. How many autosomal chromosomes are found in a normal human gamete? Okay, so humans are 2n equals to 46. You should know that. So how many autosomal chromosomes are found in normal human gametes? 22. Oh, sorry. I did that wrong. 44. Okay, 44. Okay. 1 to 22 are normal, and you have two of each. So 44. Next, how many chromosomes are found in normal human cell that is in prophase? Well, in prophase, it is still going to be 46. Okay, remember, even after you double it, it still counts as 1. So no change there. Next, how many chromosomes are found in a normal human cell during anaphase? In anaphase, the chromosomes would have been separated, so you count them as separate chromosomes, which means there will be 92. Okay? At metaphase, they all line up like that, so that's like 1, 2, 3, 4. But once you separate them, then it's 8, right? So same concept here. We doubled it. How many sex chromosomes are found in normal human zygote? Zygote is what you get after sperm fertilizes the egg. So 2 sex chromosomes. How many sex chromosomes are found in normal human gametes? 1. Right? For eggs, it's always going to be X. For sperm, it's either X or Y. How many chromosomes are found in the cells of an individual with trisomy 18? Trisomy 18 means an extra chromosome 18. So that's going to be 46 plus 1, which is 47. All right, next one. Sorry, this is 2n equals to 12. So the organism is diploid. This number tells you the ploidy. Okay, if it's 3n, then it's triploid. If it's 4n, then it's tetraploid. This is 2n, so it's diploid. And the ploidy is also number of sets of chromosome. So because it's diploid, there will be 2 sets of chromosome. How many chromosomes does the cell have? Is 12. How many homologous chromosome pair does the cell organism have? Well, it is 2n equals to 12. That means each set has 6 chromosomes. So 6 homologous pair. Okay, if you draw it out, it would look like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so homologous, 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 6 homologous pair. How many chromosomes does the gamete of this organism have? If it's gamete, it's half of the full set, half of all the chromosomes, as you say. How many chromosomes does a cell undergoing metaphase have? If it's undergoing metaphase, then the chromosome has not been separated yet. It will be 12. Okay, that is it for that. Let's do biotechnology. Okay, now I probably won't be able to finish on time, okay? But if you gotta go to campus for your math test, please go, okay? Just watch the recording later on if you need to. If you're already on campus, then good for you. What is the purpose of PCR? How does it work? If you start with 50 copies of DNA fragments, how many will you have after 10 cycles? Okay, so the purpose of PCR is to clone DNA, clone a segment of DNA. Okay. How does it work? Well, uh, basically, you don't have to describe it, okay? But like you just have to kind of know the basic principle. So for example, there is going to be primers. Primers are used to determine which part of DNA to clone. Right? They're like bookmarks in a book, right? You put 2 bookmarks between pages, and you're going to copy everything between those bookmarks. Same thing with the primers. You're going to copy everything between the primers. And we use heat to separate DNA strand. Instead of helicase, we use heat. And then we also have this thing called tag polymerase, which is for actual cloning, building DNA. Okay, because you're heating things up, you cannot use regular polymerase. They will denature, they will break down. So if you have 50 copies and then you do it for 10 cycles, that's 2 to the 10. 2 to the 10. Just plug that into the calculator. And you will get a big number. Okay. All right, why don't you guys give this a quick try? I gotta run to bathroom quickly. I'll be right back, and I'll take it up. It'll just be a minute, sorry. Okay, so let's try this question here. I'm gonna put the thing on the side. Is it possible? Nope, not possible. So leave it the way it is. Okay, we have sample number 1. Sample number 1 is treated with eCore R1. Okay, so you have to look for the recognition site for eCore R1, which is GAATTC. So let's see. GAATTC is right here. right? GAATTC. It's gonna cut it once here. And is there another GAATTC? I don't think so. So that's it. You're gonna have two fragments. Okay, you're gonna have two fragments. Number two here, it's going to be treated with SMA1, small one, which is GGGCCC. So you just look for GGGCCC is right here. It's going to get cut. I don't think there's another GGGCCC. Next one is treated with ALU1, which is AGCT. So AGCT. AGCT right here. Okay. AGCT is going to be one cut. And then do we have another AGCT? Nope, I don't think so. If you see it, let me know, okay? If you see something I don't see. Number 4 is treated with equal R1 and small one. So equal R1 is going to cut right here. And then small one's going to cut right here. Okay. And we have to load it on a gel. I'm just going to put it on the side here so it's easier to see. So sample number 1, we're going to have 2 fragments. Okay, one is longer than the other, so I'm just going to do it like this. Okay, 2 fragments. For sample number 2 is treated with small one, and I also have 2 fragments, but I hope you can tell this here is shorter than that, right? Yeah. It's not a lot shorter, but it is shorter. So in relation to this fragment, I'm going to have to put it a little bit further down. And if this piece is shorter, then this one is going to be longer compared to the other one. So it's going to be a little bit higher up. Not like all the way up here. This is a little bit higher up. Okay. For number 3, it is treated with ALU1 and the cut is kind of in the middle. So you get a longer piece compared to the other two. So I would say it's somewhere in between. Okay. This is longer than that, and it's longer than that. And then this here is shorter than the other two. Right? So that's why it's going to be, you know, lower here, and then this one's going to be higher. And finally, let me try to highlight this for you. Okay, this piece is going to be same as that piece. And this piece is going to be same as that piece because you're using the same restriction enzyme, right? So you will have the same one as this one. Okay, I'll highlight that for you. Bring highlighter on the test, okay? For you and for me as well, so I can easily see which fragments you're referring to. Okay, that one is going to be like the green guy. And then you're going to have a third piece, which is roughly, somewhere up here. Okay, doesn't really matter. Okay. That's how you put the results on a gel. Okay. Moving on. Give an example of a palindromic sequence that is 6 nucleotides long. Okay, so this is the secret. All right, this is the secret. You make up any three letters that you like, any three. A, C, T, for example. Any three you like. And then for the other three, you have to do the complementary. So complementary to T is A. Okay. Complementary to C is G, and complementary to A is T. So if you do it like that, then it will be a palindrome. Okay. Look, I'll do the other strand. You see? Same forward and backwards. Let's try that one more time in case you missed it. We're gonna make up any three nucleotides that you like. GGC. Then you're gonna write the next three that are complementary to the first three, starting with the middle one. C is complementary to G, and then GG is gonna be CC. So this is a palindrome. Forward and backwards are the same. Understand? I hope so. There are three different DNA samples are treated with equal R1 and separated using gel electrophoresis. What is the purpose of the marker laying? This is the marker laying. It is a DNA letter used as a reference for the size of each band. Okay, I just received a question. There's a question about sticky end and blunt ends. Should go back to here. In this chart, if it cuts zigzag, then it's a sticky end. If it cuts straight down, then it's blunt ends, right? So the advantage of having sticky ends is if you have multiple DNA samples and you treat it with the same restriction enzyme, then they will create complementary sticky ends. Okay, so remember if we have the plasmid and then we have the DNA, we have a gene that we try to insert it in. If we treat them both with equal R1, then they would have complementary sticky ends, meaning the sticky ends that you get from here is going to be able to stick onto the ones that you have in the plasmid. And in doing so, you are able to insert the genes into the ring. Right? This is not possible if it's blunt ends. If it's blunt ends, they do not connect like end to end like that. Okay, it has to be there's some kind of overhang we call them, and that allows them to stick back together. Did you answer your question? Or yeah. Beside SMA, no, this is also blunt. These two are blunt. You see, it cuts straight down, right? Okay, for it to be sticky end, it has to create like a little L shape. Okay, there has to be some kind of things that are sticking out on its own, as opposed to like a clean straight cut. It's a zigzag cut. Okay? All right. Which sample has the longest DNA strand before being treated with equal R1? Okay, so this one is 1,000. You can tell from the ruler it's 1,000. This one is 800 plus 200, right? 800 plus 200, which is also 1,000. This one is 600. You just read it off, right? 600 plus 300 plus 100, which is also 1,000. So this is a trick question. They are all the same. What sample was not cut by restriction enzyme? A. Because you only see only one fragment, only one band. Okay. Explain what will happen if you make the following mistake while doing electrophoresis. Forget to load the dye. If you forget to load the blue dye, then the sample, likely the sample will not sink into the well. Okay? Some or all sample will be lost. And I have another question for 4B. Uh, this was 4. Again, never mind the numbering. Different line is okay. 800 and 200 different line. Yeah, I mean like I'm not 100% sure if I understand the question, but let me try, okay? So over here, this line, if you go to the ruler, it says 800, and then this line over here, it says 200, right? And this length is one sample, right? So if you add them up, it works out to be 1,000 before you cut it. Right? Okay, great. All right. If you reverse the positive and the negative terminals, so remember when you do gel, the top is negative, the bottom is positive, and the current's going to go this way, right? But if you reverse it, then the current's going to go up, and all your samples started up here in the well. So if you run the current backwards, instead of going down the gel, the sample is going to go up the gel, right? If it goes up the gel, then you're going to lose the sample. Right? All samples will move up the gel and be lost. Left the power supply on for too long. Okay. If you run the gel for too long, then again, all samples will leave the gel at the bottom. Right? Think of that playground structure I showed you. When given enough time, even the adults can make it all the way to the bottom and exit. When given enough time, even the long DNA, it's going to be able to weave itself through the gel and exit at the bottom. And then you will not see anything. Forgot to add ethidium bromide. Ethidium bromide allows you to see the DNA bands when exposed to UV. So if you forget to add it, cannot see the DNA sample. Okay, so if you do the experiment and you see no band in the end, you actually don't know which one of this is the problem. Okay, like did you forget to add the dye? I mean, you can know, you will know because your gel will not have the blue line. Okay, did you reverse the terminal? Did you left it on for too long? Did you forget to add ethidium bromide? So like these are all things that could go wrong and cause you to not see any DNA band in the end. We did this question in class, but very quickly, which sample will be further down the gel? This one is going to be further down the gel because it is shorter. Shorter DNA always will be able to move further down the gel. We also did this in class, I think, right? The plasmid below is treated with equal R1 and BAM-H1. So you cut here and you cut here, and it's separated with gel electrophoresis. You will have two bands. Okay, one band. Uh, this would be like a lower band because it's shorter, and then this here would be like a higher band. Next, a gene is inserted into the same plasmid shown in question 8 using E core R1. The plasmid is transformed into the E. coli. So it's pretty much same stuff that we've been doing in class. And you're gonna treat it with tetracycline. Tetracycline. Okay. So is it going to survive? Well, if you insert with equal R1, the gene goes right here, and it will not interrupt the tetracycline resistant gene, so you will expect some growth. Since TETR gene is not interrupted by the insertion. However, if you use small one, SMA1, which is right here, right, that's the SMA1, that's the restriction enzyme site, what's going to happen? Well, it will be no growth. No growth. All bacteria will die, regardless of whether it accepted the plasmid or not. Okay, if they don't accept the plasmid, they will die, of course, because antibiotics killed them. But even if they accept the plasmid, it's not going to help them because we decided to use SMA1 to insert the gene, right? Which means the gene is going to go right into the middle of TRTR, TETR. So it's not going to give them resistance. Okay. SMA1 insertion will interrupt TETR gene, not giving them tetracycline resistance. Even if they even if they accept the plasmid. Okay? For more practice on these kind of stuff, look at the file that I post, right? After lecture 9 with the extra plasmid practice. Okay, we're almost there. I think like maybe another 10 minutes we'll be done. Body tissues. Label each of the following cavities. I mean, we did it already in class, but just go through it again, I guess. This is cranial cavity. Okay, it has brain. Okay, brain in it. Just know the ones I'm labeling. Okay, this is thoracic cavity, which has things like heart, lungs. Okay. Abdominal cavity. You guys gave me a bunch of stuff in class already, okay? But stomach, liver, intestines. Okay, like I said, I'm not gonna give you weird things on the test. It's just things that you probably already know. This is the pelvic cavity, and it contains things like urinary bladder, right? Uterus. Okay, number two, we did a bunch of these things in class. Just go through all those slides again on your own, and you should be okay. All right. um, on top, below, further away from the middle, closer. Just review those slides, and yeah, and you will be okay. All right, very quickly here, last one. The yellow plane right here, that's the one that will cut the body into anterior and posterior portion. We call that coronal or frontal plane. Okay, anterior, posterior cut. We're gonna have this blue one right here that cuts it into top and bottom, transverse plane, superior, inferior. And then finally, we have this one that cuts it into front and left and right. That is sagittal. Sagittal plane. Left and right. And that's it. Okay. I'll see you all on Wednesday. Good luck with your math exam today and your chemistry tomorrow, I think. And be on time on Wednesday. All right. If you have any other question, feel free to email me. Take care, guys. Drive safe. Bye bye.